Hello, everyone. Um, I'm just really clicking a lot of times to accept everyone in the meeting, as this is in the process. So, um, thank you for your patience. Okay, so the first batch is in. Hello, everyone. Nice to see you guys, to see familiar faces. Um, Hello, Ivan.
Hi.
Hello, Ken. Okay, so still new people. So there have been, I think, 30 people who have signed up. Not everyone shows up though, but yeah, I think there's going to be quite a number of investors, potential investors. And just for you guys to know that the meeting is recorded, and we are going to share this with the rest of the community of investors, the ones who, because of different time zones or busy schedules, did not have the possibility to attend in real time. And the meeting is going to take 1 hour. For the ones who have already attended, they know what the schedule is. So first of all, let me spend a couple of minutes and introduce the 2060 Foundation, what we're trying to do, and the whole context. So I'm going to share my screen and and get going. Okay. So the 2060 pitching session, this 2060 pitching session is an initiative of the 2060 Foundation. What we try to do is help humanity to the best of our abilities to defeat aging before the year 2060. So a couple of things about the 2060 Foundation. First and foremost, it's a nonprofit. It's very important for you guys to know we're not looking for profit in this organization. What we're looking is for additional years of healthy life. We have more than 200 members, some of them very active, like the core members, and some of them getting involved here and there when they have some time. And our flagship project right now is the 2060 Longevity Investment Club. So what we try to do is bring as much capital as possible into longevity, um, and show the rest of the world what a great promise longevity is in terms of science, of business, of startups, and so on. So a couple of things about our team. So our core team is composed of some of the people that you see here. I didn't have the time to update the team. There is new members who have joined recently, but the most prominent ones are Manish. You've maybe heard of him. He has worked for the Buck Institute for many years, and now he's a general partner in an investment fund. And Max Kamrani, who has founded the 2060 Foundation with me. And of course, as many of you may already know, Brian Kennedy, who is in our advisory board, he has been director of the Buck Institute for many years before joining the Singapore University, where he's a professor in the departments of biochemistry and physiology. So the meeting today is going to have two components. We have two startups we're pitching today. The first one is going to be Cortex Discovery. And Ivan is going to pitch, and he's going to have 10 minutes to pitch and then 10 minutes of question and answers. And then we have Life SE by Adi Diaz, who is going to come second, same stuff, 10 minutes of presentation and 10 minutes of question and answers. The whole meeting is supposed to last no more than an hour. So yeah, that being said, let's get going and start by introducing Ivan and Cortex Discovery. So Ivan, please, you can share your screen and yeah, the floor is yours. And you have to unmute your mic also.
That's done. It's not the usual one. Screens. Okay. Two. Okay. Allow Zoom or to share screen. It's not the usual.
We still don't see your screen.
Yeah, I know. Okay. I have to leave the meeting and come again because
Okay.
it's a different. So I'll be there in 10 seconds.
Okay.
Okay, now it should work. Do you guys see my screen?
It does. Yeah, absolutely.
And do you see my second slide?
We don't see your slide. We only see the your Chrome browser.
Oula. Uh, so sorry, because that's not the usual. Wait a sec. Okay, I'm going there. Now you're good, right?
We still see your Chrome browser.
Sure.
Oh yeah, no, it's good. You're good to go.
Good. Do you see the second slide?
Yes,
Good.
yes,
Perfect.
yes. I
Okay.
prefer you on the left side than on the right
Okay.
side.
So let's start from the beginning. So I met my business partner about 36 years ago. We were at 10. I told him I wanted to live forever, and he replied that he would rather replace humankind by robots. So 15 years after I got my PhD in the field of aging genetics, and I had some targets in mind that could drastically extend lifespan. But when I started to look at the landscape of drug discovery, I got a little depressed because of the drug attrition rate and the cost and the time that it takes to make a drug. So I started to focus on cheminformatics to accelerate the preclinical part, that is the discovery, let's say, phase. And there I started to realize it was mostly chemists trying to do mathematics. So I contacted my old friend who was back in the time professor in quantum information. And I convinced him to retire and invest 1 million in the project that became Cortex Discovery. We've gathered most of the publicly available data about compounds and their properties. We have about 750 million data points. We then created some novel neural network architectures, some graph neural nets that correlate the molecule structure to a property. So it's like raw data. We have the smile of the compound, and we have a binary output, active, inactive, or a number, how much is it active, or a dose response curve. It can be anything. So activation of a target, inhibition of a pathway, number of cells that die, is it soluble, toxic? So some of the features of the neural network is that it predicts everything at the same time. So 5,000 properties for 10,000 molecules per second. It's therapeutic field agnostic. And we saw something that was astonishing for us is that it looked like our predictions were as accurate as the reproducibility of the experiment, which is unprecedented in the field. So we needed to do some actual physical proof of concept. So this was during COVID, and that proof of concept was something you might smile at, but I convinced a professor in Aarhus University to test only 6 compounds for a repurposing project. So we had some data regarding assays, so experiment testing compounds on SARS-CoV-2 infected cells, but that was not enough. So since we have this multi-parametric prediction, we started to explore the possibility of using it. So that led us to create that system that comes after the neural network, and that allows us to instantly filter millions of compounds based on their properties, like the probability of activity, potency, and efficacy, and also control for false positive, such as fluorescence, toxicity. So we looked at compounds, for instance, directly acting on viral protein or on cell protein that are being hacked by the virus. To make the story short, 5 out of the 6 compounds worked at non-toxic dose. 2 of them worked before and after infection in 3D human lung cell cultures. There are patent pending. We published in antiviral research the basics of the neural net, but not the full structure, of course. And we have also done something similar with Stanford University, and we will do a follow-up publication. We were curious about the accuracy of our predictions, and so we had a collaboration with the Victorian Center for Functional Genomics, where they gave us an external training data set, actually 80% of it. It was a high content screening imaging data regarding they were looking at compounds selectively killing cancer cells. And we were testing on the 20% remaining compounds, and we proved that we were as accurate as the experimental reproducibility to predict compounds killing these cell lines. So now let's jump to the business. So the pharma companies would usually screen about 500,000 compounds for a specific target for about $1 million. And what we can do is reduce that screening at 10 times less, actually 50 times less easy. And but if it's 10 times less, we can then make a calculation, and we have done such proof of concept also. And we would screen virtually all the physically available libraries from reliable providers, which we have. It's about 23 million compounds. And if we follow the retesting processes, we would have about typically 5 times more hits. So the cost efficacy here is about 20 times improved. We can also predict simultaneously the ADME-tox properties, so the pharmacological properties, solubility, permeability, toxicity, inhibition of key enzymes such as cytochromes. And this is automated, so it's ready for software as a service. We have organized our compound libraries per approval speed, because we want to have fast access to longevity compounds. And I actually already started to eat some of the compounds that come out of the neural net. And so we are classifying the libraries by what's approved nutraceuticals that can be into pills this year, natural compounds that are food-like or drug-like that could become novel nutraceuticals and bypass the whole FDA thing. Existing drugs or drugs in trials for repurposing, drug-like compounds that are physically ordered usually by pharma or biotech companies, and virtual libraries. So regarding virtual libraries, we can span trillions of synthesizable compounds with our new generative AI system that enables us to span quite well these chemical spaces. So the business model is mixed. If we offer services individually, so that's typical service that are offered in the preclinical phase, like high throughput screening for specific target, pharmacological properties, so the virtual ADMET, the side effects, the off target, and also tweaking a lead that is maybe too toxic or not potent enough. and that is typically as accurate and 10 times cheaper and faster than what you would do in a wet lab. And we have that other more interesting possibility where we propose lead compounds, but there we parallelize the heat discovery with lead optimization, and that would lead to something that's in the range of 100 times cheaper if people were doing it from the beginning till the end. And then we would be paid by milestones like active compounds, if the compound is non-toxic, a little bit more, and then we agree on some properties for another milestone, then the IND, and then the different clinical phases. In parallel, we are also developing our own pipeline of longevity compounds. So the deal usually is that we are producing the in silico leads. Our partner is testing the compounds in vitro or in vivo for free. We share the IP for patents, publications, look for money for grants, and that will ultimately lead to spin-offs in different types of molecules. So we've been very successful already, and I'm proud to say that we're the first AI company that has a compound that comes out of a neural network that extends the lifespan of a full organism. We did over 33% for C. elegans. We also discovered three novel NRF2 activators. It's a master regulator of most key antioxidant enzymes. We have four novel mTOR inhibitors. That's with Brian Kennedy, by the way. And we have seven novel DNA repair enhancers that with Vienna University. We should have more of a master control of DNA repair that will be tested in a collaboration with the NIH. We are also doing a senolytic project with them, and we will keep the IP there. We have some sort of deal. We're also working on projects of reprogramming and age-related disorders. Of course, like we are keen on starting producing nutraceuticals, and we joined the team for the X Prize also. So it's quite important to note that all these compounds have been discovered in the last 8 months. So that's probably 10 to 15 years of work in a laboratory that would have something like 20 to 50 times more money. And then the cherry on the pie, we have a docking prototype, and that's for targets for which there is no experimental data. We will not learn from crystallographic data, but we will learn from simulation, and we use deep learning to compress the information. So, I mean, we already do it at the moment, not at the quantum level, but we have a prototype that's as inaccurate and 1,000 times faster than current system. And once it will be tweaked properly, we believe that we can basically replace most of preclinical experiments by combining our two tools. So we're looking for 10 million euro to cover our costs for about 2 years, and then at this stage, I think we can lead the field of preclinical drug discovery, AI guided, let's say. Our team is made of mostly experts in the field of quantum physics, AI, and programming, chemistry too. And we are backed up by world-leading scientists in the field of longevity, such as Emma Tilling or Breda Gray. And now we're just looking for talking with investors because I think we are ready.
Thank you, Ivan. Thank you very much. I'm really delighted to hear again from you. We've discussed a couple of months ago, and you still made a lot of progress, so it's exciting. Here comes the question and answer session. So if anyone has questions, please go ahead. I have my questions, but I will let you guys start. Okay, if nobody wants to ask question, I will start asking mine. Oh yes, sorry, Carl. Of course, please go ahead. You would have to unmute your mic. So
Okay, sorry about that. Thanks, Ivan, for the presentation, and Gabrielle for the time here. One of the things that, in the research development, can you kind of focus more on, from research to launching the startup? I mean, what are some of the steps that you're planning to do in that 2 year time for that $10 million?
So actually, we already started to have customers, not too many, but I'm negotiating with many. So the idea would be to really sell the service to biotech and to the pharma industry. But I would focus first on longevity startups because we are curating the database, and part of the money will be linked to the curation of the database. The software as a service, it would be just for the ad metocs, and that's something in which we don't invest. So we have the possibility of generating this automatically, but we don't have the interface, and that's something I think would be worth just to, let's say, make some noise in the field and attract some more like pharma customers. And regarding the longevity pipeline, I've already been discussing with companies in the field of senolytics that would actually buy my compounds and pay me more at different steps of evolution towards the IND. And we would also work on the docking, and the docking, we're still open. It could be software as a service, but if we are good enough and can cut some deal with the pharma industry, we could have some exclusivities like for them on specific targets, because technically we would be able to produce leads in most therapeutic areas because we have most of the therapeutic targets in our database.
Okay. Yeah, I would like to talk to you more offline, after this event. So appreciate it. Thank you.
Thank you.
Okay, next question. Okay, I will ask mine then. So what do you think, Ivan? What's your take on the recent OpenAI module for longevity modeling and using AI for drug discovery? And maybe there will be other ones. So what do you think of your competition?
So at the moment, I don't see that as a competition because language models are very different. So we use graph models. So we really look at the chemistry. So it's a different. I mean, so I would expect a question rather regarding AlphaFold, for instance. They are getting a little bit closer, but it's also different because they are really good at predicting the protein itself. So we will use it actually for our docking, but so far their technology is not really useful, and we don't see how they will be good at predicting really drug target activity because they don't really look at the binding. So there is a little bit of an effort to work with force fields, but it's not exactly. So it is not a competition at the moment. Regarding the language model, actually, I would probably use something like DeepSeek in order to accelerate the curation of our data. That's possible. We are already using a little bit of ChatGPT to accelerate some of the analysis that have to be done manually. So things are becoming very automatic, but once we have a new target that we have not seen, someone has to go, through the experiment and just double check that we know in which direction the signal is going and what are the counter screens and so on. So far, I see these tools as complementary to ours, and they will enable us to be faster, I guess.
Okay, interesting. Thanks. Another question I have already asked you a couple of months ago, but now it's a different version. When we look at the what happened with ChatGPT, so when they released this a couple of months, or let's say 6 months or a year later, there were many other tools that were similar, and there was a huge competition, and that was good for the world, but still. What makes your solution so special, and how can you protect against future competitors that could come and do the same thing?
Okay, so actually, I think it's special in the sense that that I don't think that any of the competitor has such a technology, because especially the big ones, because they would actually make money as selling that as a service. And I've been, for instance, discussing with with the Alexei Zavaronkov, in like the founder of InSilico. They are probably the biggest competitor, and they have some high throughput screening capabilities, but they don't use it to do that deep learning based QSR that we are doing. That's what I would do with the technology that we have. And they don't do ducking either. So I have the impression that so far the companies that focus on drug discovery and AI are probably tweaking a little bit some off-the-shelf tools, but I don't see anyone that has a technology that works. But then you never know because there are smart people out there. We can protect the compounds that we find, but I'm anti-compound because we've been a little bit fighting with chemists and the pharma industry because they think that compounds are important, but what's important is rather the algorithm to find them. And yeah, that there might be at some point some people doing something similar. But regarding the first tech that we have, we reached the pinnacle almost, let's say, because we are on par with the experimental accuracy. So I don't think they can do much better. So we will focus on longevity also, and we also have some targets in mind. So we have a little bit a few tricks that would enable us, even in such situation, to still lead some niche, especially in the field of longevity, which is our main focus.
Interesting. Can you give us some numbers about the valuation and the conditions uh, according to which you're trying to raise?
Yeah, so we're looking for 10 million for valuation pre-money at around 30 million. It might increase shortly after the result we will have with the Senolytics, because regarding the predictions that we have, we might have compounds that are killing cancer cells and that are highly senolytic, and we should also close some deals. So I mean, I couldn't speak about everything in 10 minutes. So, but we have ongoing things. So we are, for instance, not profitable in terms of customers, but in terms of what we have raised from grants plus customers, we spent much less money in the last 6 months than what we gained. So we just really need to. I think it's the time for us to scale up, because there are lots of things that we could do and that we're not doing because we're just simply 4 people, like full time, plus some couple of people that do some business development on commission basis. So.
I understand. Thank you very much for your clarification. So 10 million on a valuation of 30 million,
Yeah,
pre-money, I guess.
yeah.
Okay. That I think it's a good conclusion. Time's up. So thank you very much, Ivan. Some people will reach out to you most likely. Maybe Carl, who has already mentioned it, but also others. And again, there's going to be other investors who will watch this offline from the community and get in touch with you.
Yeah.
So let's go. Let's get to the second startup, which is Life SE. Adi, maybe you wanna share your screen. Ivan, maybe you can stop sharing your screen
Yeah.
and give the lead to Adi.
Yes. Let
Thank
me
you very
try
much.
to do that. Thank you.
There you go. Do you hear me well?
Yes, we do. And we see your screen also. Perfect.
Fantastic. First of all, thank you for the opportunity. I'm Ari. I'm the founder of Life SI, and we work towards creating human tissue and organs in the lab. The main goal that we have is to do it for patients that are right now in the waiting list. And every day, 17 people will die only waiting for an organ in the US. And every 8 minutes, a new person is added. Even though our main goal is for patients in the waiting list, this could be used for longevity, right? When your organ fails, you can work creating a new one. So how the process works? We take a CT scan or an MRI or any scan from the anatomy of that specific patient that needs the organ or the tissue, and we take a small biopsy of his or her own cells. Combining the geometry from the CT scan or MRI with the cells from the biopsy, we can create the human tissue in the lab, replicating the anatomy or even creating a new kind of anatomy for that specific patient with their own cells. So this tissue can be used for the main goal that we have, that is going back to a patient as the implantation for patients in the waiting list, but also can be used for testing new drugs. Combining with the previous startup from Ivan, we can test those drugs in more realistic models instead of using animal models that we know that they don't represent what's going to happen in the final patient, or traditional cell culturing that we know that is not realistic. So the main goal of this is no waiting list and no immunosuppression. So the patient is going to get their own cells, so they don't have to go through immunosuppression to prevent the rejection of the new organ. So this is not only a nice project with a humanitarian impact, this is also a huge market. We already identified the transplant centers in the US. That is our main market right now. And every year, more than 45K transplants are happening in the US. So it is a market that, even though if you can solve this problem for all the people that is in the waiting list right now, the next year there's going to be more people. So it's a never-ending market. So right now we are in our seed round. We are working towards the creation of a fully automated platform for creating these tissues in the hospital, and also doing the proof of concept using cartilage stuff from a real patient. The next step is going to be working on skin. Right now we are working on cartilage. Next step is going to be working on skin, and the next one is going to be for more complex organs and tissues. We already have prepared a spin-off company for the next round that is going to be only focused on testing drugs on these tissue models using cells from patients that have the pathology that we want to research on. So that is the main goal for 2027, the spin-off of this company that is already incorporated also. The investors that get in this round, they're gonna get participation in that company also. So right now we are focusing on microtia. Those are patients that born without the ear or with malformations in the ear. So right now the solution, the surgery is taking cartilage from the rib cage, and they do manual work in the operating room. Instead of that, we are working on creating from the CT scan a replica of that ear for that patient using their own cells. So we have a very interesting team, a young team, but they are very passionate and they are nerd, everyone in their own field, biotech, biomedical engineering, mechatronics. And I've been in this field for 10 years now. I'm an invited professor in different universities for teaching 3D bioprinting and medical images processing, even though I drop out med school, but I keep getting these invitations to teach in different universities. So we have huge advisors. One of them is a surgeon that is going to move forward with the actual implantation. That is an expert in microtia, especially. We have people from immunology, business development, and we already have all the plan for scaling up this project in the US. So this is not only an idea, we already have products in the market. We develop and manufacture fully in-house 3D bioprinters and the consumables for these 3D bioprinters. We already sold 14 last year. This year we are continue selling, and right now we are focusing on the US market especially. So these are our facilities. We are not working inside a university. This is a fully private lab that we have for biomaterials and tissue engineering. and all the manufacturing of the equipment that we develop in-house. So we already got the approval for using tissue samples from patients to get the tissue samples, isolate the cells, and work with those cells to create the tissue. And we are also working on vascularization. This is huge because all the models that you have in the market, they don't have vascularization, actual vascularization. You can have channels, but there is no vascularization. So we are working on that because there are some drugs that work on the vascularization of the tumor, for example. So if your model has no vascularization, it doesn't make sense to test the drug on it. So we are working towards that, both for the modeling of the tissue and actually, and mainly, for making the tissue viable for implantation. So right now we are focused on replicating the ear from a CT scan of the patient using the patient cells. And we are focused on that. That is a very simple application with a small market at first, but we need to make the proof of concept that we can automate, and we are doing it. We are automating the whole process of fabricating the tissue using the patient cells. So this has another applications for replicating tumors, for example, and testing different approaches for that specific patient, and then go to the actual patient and try the drug that we found out. So how the business works, we sell biofabrication platforms and the biomaterials, the consumables, to hospitals. While now we are selling mostly to universities and research institutes, and we are moving towards hospitals. And they are using it for research mostly, not only for creating tissue, but actually in some cases for creating pills, for combining different drugs for specific patients in the same construct, and yeah, doing by 3D printing. So we have competitors, of course, because this is a huge necessity, and it, yep. I don't know, Gabby, it's okay?
No, sorry, I just coughed. I should have put my mic off. I'm sorry.
It's okay. So the main differential that we have with our competitors is that we are focusing not only in 3D bioprinting, that is only creating the tissue, and that is only one step in the whole process of fabricating tissue for patients. So we are automating the whole process. And that is very important because if you want for this to be the gold standard for patients in the waiting list, you cannot go to a hospital and say, all right, I'm going to install this bioprinter, but you have to build all the infrastructure, train people, buy all the equipment that you need. Usually, the hospitals are not going to have a tissue engineering lab. So we are automating like a on-bench platform. So like the black box, they put a little tissue sample and then get the actual tissue for that specific patient. So we have two main milestones. One of them is to get to the preclinicals of human cartilage fabricated in our lab. And the second one is the MVP of the fully automated biofabrication platform for using in hospitals, not for research, but actually for doing the tissue or creating the tissue for specific patients. So right now we are raising $1 million for the next 18 months using a site with a $6 million post-money valuation for these specific milestones for the next 18 months. We already raised 275, and we are trying to close the million in the next 1 or 2 months. So thank you. Thank you very much for the opportunity. I'm very open to talk and answer all the questions.
Okay, thank you, Adi. Great presentation, great project, and great prospect of what medicine could become in the next years with such a technology out there. So, who has questions? Okay, let me be the icebreaker then. I have two questions. You have discussed about competitors, but there's there's two that I haven't seen on the screen on your slide. One of them is the United Therapeutics, Martin Rothblatt, who is printing as far as I know, lungs, and they are getting more and more precise into doing that, which is a necessity for this organ. And the second one is Doris Taylor, who has been working in transplantation of hearts, ghost hearts. What's your opinion on those two companies? And I know they were pretty advanced.
Yeah, they are. There are another companies also, and even companies working on human cartilage for a year. But the main difference is the focus on doing or creating the tissue for going back to the patient. Most of them are starting in modeling the tissue, and a lot of them are working on real complex organs like the heart, for example, that has not only specific tissues inside, but actually a very specific function. So you don't you need the geometry to be perfect, the communication between the cells, and the function of pumping is not easy. So right now we are focusing on things that are realistic because we like to work in that way, right? So mainly, maybe our pitch is not as fancy as others, but we are very realistic on the things that we can deliver and when we can deliver it. So that is why we are more conservative, but more realistic on the results. We already have products in the market. We've been working on this for a long time. We have our own tissue engineering lab. So that is why we are a little bit more conservative, but more realistic on the results. And our dynamic of working on is creating revenue streams all the way that we are working on. So that is why we work with cartilage, and then we get revenue from cartilage and move to skin, and then get revenue from skin. That is why we are working in that way. The main goal for our company is working on kidney. There is the long-term goal, because 80% of the transplants are from kidney. But when we present, I like to present the things that we know that we can deliver in the time that we are saying we are going to do.
Audie, I have a
Thanks
question.
for clarifying. Yes.
I'm Terry O'Malley. It seems like now your focus is on skin, and you also brought up the subject of cartilage. Does this application also apply to skeletal cartilage, joint cartilage, skeletal, spinal cartilage, those types of issues?
Yeah. Right now, we are focusing in the first step of using the cartilage for aesthetic applications because of the risk. And then we can work on a joint cartilage. But the main goal right now is only for aesthetic applications in ear and nose because of the risk. So we want to get to the patients as fast as possible. And from that, start building more complex applications or tissues like skin, or because of the vascularization, or another applications of the actual cartilage. But right now, only in aesthetic applications like ear and nose, because of the risk and the short-term application that we can create with that.
What's your timeline on delivery of the cartilage?
We are trying to get to the preclinicals within the next 18 months.
Okay, so a couple of years. Okay.
Yeah. For the clinical, we work in parallel with the other tissues, but the proof of concept, we want to do it with cartilage. In the clinical application, we've been a lot of times in Prospera. I'm not sure if you know it, Vitalia, Prospera. There is a place, an island in Honduras. I've been there from the start, and we go. Most of our investors are from the longevity field, and there is a possibility of making the clinical trials in there in the shorter term than doing in the US.
And what would be your delivery or your application or production here in the United States?
Mostly the manufacturing of the equipment and the actual tissue for these applications. Depending on the tissue, the business model is better to work as a service. So they send us a small tissue sample and the CT scan, and we create the tissue and deliver the tissue. And for more complex organs, the best way to do it is to install the capacity in the hospital, in the transplant centers. So that is why we are working in the two sides, developing the equipment for the hospitals and developing the research needed to deliver the actual tissue.
How about a lot of the pain clinics here? I mean, that's a big business here in the United States. And right now, the pain clinics.
Oh, yep.
Pain therapy. That seems to be a very fast growing, part of the medical industry. And right now the application is more stem cell therapy than any cartilage replacement. So
yeah. Actually, part of the process can be used for that kind of applications because we isolate the cells from a small tissue sample and we replicate the cells in the lab. So automating that process not only help us to create an actual big tissue from a small tissue sample, but actually for mass cell production from a small tissue sample or a small vial of stem cells from the patient.
Okay, thank you. I appreciate you. You're on the right track, that's for sure.
Thank you. Thank you very much.
You're welcome.
Ivan, you had a question, I guess?
Actually, Terry's question was covering my question.
Okay, I have a practical question then. Why do you need the CT scan for disease in the ear? Because if the ear is already in a bad shape, you don't want to reproduce the same one. You want to have like a normal ear, so you don't need a CT scan to reproduce something that anyway is not accurate.
That's right. Most of the times, actually, I think it's over 80 or 90%. It's only one side. They have one healthy ear. So you can use a 3D scanner also, a manual 3D scanner instead of the CT scan. But we can replicate and mirror the image or the geometry from the healthy one and replicate it for the not healthy one.
Makes sense. Thanks.
Hi, I've heard presentation on, producing tissues and stuff, but I never get a chance to ask, how's the texture of it as it's man-made? What's the texture? How is the elasticity? How's the feel from a real, like a real ear or nose or whatever?
Yeah, as we are using the same materials that build the tissues naturally, like collagen mostly, and we are using actual human cells, the consistency of the material is very similar to the actual tissue. Depending on the cartilage, you have different consistencies. Most in the joints, there are more rigid and they are more flexible in the ear or the nose. So combining the biomaterials, that is why we have one of the biomaterials lab and the other one for tissue engineering, because one of the key things for this is creating the material for building the tissue, for scaffolding the tissue. So we already have developed some. We sell those for research, and we are focusing right now on developing the specific one for cartilage. But the consistency is very similar to the actual tissue. And we usually use collagen combined with different things, but naturally our tissues are made out of collagen with the specific cell of the specific tissue, but collagen is the main matrix.
Thank you.
Buddy, I have one more question. How
Yeah.
does it react how does the tissue react to sunlight, UV?
We didn't try that with the skin model that we are working on. We are working on epidermis, the outer layer of the skin. We didn't test with sun. It should work like the actual skin because we use keratinocytes. Those are the actual cells. But we didn't test it with the sun. We could try it. We did try it with different cosmetics. So there is a short-term application of human skin, testing cosmetics instead of using animal models. And that is aligned with a cruelty-free movement. But we didn't test it with sun. We can try that.
How about neural activity? Like if you get cut or you damaged or you got hit or anything like that, is it a dead zone or is there actually any sensitivity in the ear once it's replaced?
It actually depends on the lesion or the harm of the tissue. But usually what you do is to implant the new ear under the skin. You do like a pocket and you put the new ear under the skin. So the skin still has the sensitivity and the neural termination. Yeah, that's right. And the vascularization that is going to keep alive that tissue that you are implanting.
Okay. All right. Thanks.
Thank you.
It's very interesting. What about the reproduction and differentiation of stem cells? As far as I understand, this is where your tissue comes from. Do they turn into cancer cells or carcinomas?
I didn't get the last part.
Yes, my question is how accurate is your technology to can you hear me?
No. No. Yeah. So I get it.
Okay. Let me repeat. So I understand one of the first steps of your technology is to take cells from the body of the patient and then multiply those cells and differentiate them into cartilage, right? So my question is, what are the risks of those stem cells turning into cancer cells or carcinomas?
Yeah, that is a great question. We actually try to not use stem cells because of that. We use already differentiated cells. So the biopsy that we take from the patients is a biopsy of actual cartilage. And we isolate those cartilage cells and then reproduce them. We could work with stem cells, but you have to differentiate them before you go to the actual fabrication of the tissue, because of the mechanicals of the process can affect the differentiation. So you can get cells differentiated to cell types that you didn't want only because of the mechanical forces that we have during the fabrication. So that is why we try to work with always differentiated cells. So we take a small tissue sample from actual cartilage and then use those cells. You could work with stem cells, or even you could work with fat tissue cells and then de-differentiate them and create with induced pluripotent cells and then differentiate it to the actual cell type that you need. But right now we are trying to reduce the risk to get faster to a patient. We could do it with stem cells, but it's risky to get to the patient.
I understand, but if you take a small sample of the cells of the patients and then you divide them, then maybe the telomeres are going to shorten very quickly. Oh, you lost me. I'm going to repeat. So my question.
Let me try with.
I don't know. Maybe it's because of me. I don't think so. I have a very good internet connection most of the time, but it could happen. Can you hear me now?
Yeah, perfect.
Okay, so let me repeat. So if you multiply the cells that you have extracted from the patient's body, does this mean that the telomeres are shortening and the sort of tissue that you have is going to be sort of biologically older than it used to be when the cells were extracted, and that it would get older quicker, faster?
We didn't try that. We didn't measure that. it should grow at the same pace that it does in the body. It shouldn't change unless you promote the fast growth of the cells using bioreactors. But we didn't measure, so I cannot answer you.
But every time a cell divides, the telomeres get shortened, right?
Yeah, but as happens in the body also. So it's the same process as in the body, but in an incubator.
I understand, but when you take a small sample of cells of the patient's body, then you have to multiply them, and you multiply them pretty quickly because you want to get to the threshold of tissue that is going to make the ear, right? And when you multiply those cells, aren't they getting older faster than they are getting old in the patient's body at a slower pace?
I cannot answer that. I'm not sure. I don't have the. Could happen, but I'm not sure. It is a great question. Yeah. If it happens,
thanks.
It would be useful for creating agent models.
Yeah. Okay. Thanks. Is anyone having an additional question? Gonna be next problem. Okay. There's gonna be the next problem to solve with a complementary stem cell startup. Yes. Okay.
Yeah, for sure.
Great insight. Okay, so one last question if anyone has anything to ask, and otherwise we're going to wrap up.
Not exactly a question, but a suggestion. In case you do determine that the telomeres do shorten during the expansion stage, there's a company, I'm posting it in the chat at the moment now, that's developing a telovance therapy to extend telomeres in the ex vivo expansion stage specifically. So might be of interest potentially.
Yeah, for sure. Thank you. Thank you very much.
No problem. Thanks for the presentation.
Thank you.
Yeah, guys, so time's running up. We have to wrap up. Thank you very much for everyone to be here. The projects were super exciting. I really hope that investors are going to reach out to you as a consequence of this pitching session, and you will be able to have the necessary resources to bring your projects on the market anytime soon. So thank you very much again. I'm most likely going to follow up with you also and do some intros to other investment funds that are looking at those kind of projects for potential investment. So thank you again, everyone. Bye-bye.